everyone, and welcome to today's webinar, Improve Ergonomic Safety in Your Lab, Reduce Pain and Strain During Pipetting. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Sartorius. To learn more, please visit sartorius.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers. Mira Voutilainen, Product Manager, Liquid Handling Consumables at Sartorius. Nadim Mira, Product Marketing Analyst 2, Liquid Handling Automation Solutions, Waters Corporation. And Jasper Eaton, Senior Product Marketing Manager, Liquid Handling Automation at Waters Corporation. We also have Vanis Van Holm as a guest who will be interviewed by Jasper. So Maria, Nadim, and Jasper, you may now begin your presentation.
Hi everyone. So let's get started. In my part, I'm going to be discussing about laboratory ergonomics, and I am especially going to focus on how pipetting affects ergonomics and how it can be improved. But let's first begin with the very basics. So what is ergonomics? The short definition of ergonomics is that it is a science focused on designing and arranging things people use so that the people and things interact in a most safe and efficient way. If you think about a laboratory as a work environment, you can probably quickly pinpoint several ergonomic challenges that you face from day to day. It is characteristic for lab personnel to be working in a very static and awkward postures, like when pipetting or when you, for example, need to use a laminar flow hood. Moving heavier objects such as different equipment, water, chemicals, you name it, will also create musculoskeletal load throughout the day. There is also a lot of repetitive work in pipetting and in opening and closing tubes, just to name a few of the most common tasks. And often while doing these things, we tend to lean on tables with our elbows, which creates contact stress to elbows that are not exactly designed for supporting weight. There has been a lot of studies about the amount of musculoskeletal symptoms that lab professionals experience, and the numbers are quite high. Up to 60% of laboratory personnel do suffer from different musculoskeletal symptoms. These symptoms can, for example, be pain, weakness, stiffness in arms and hands, and this can also lead to decreased range in motion. Maybe the largest contributor to risk of developing different musculoskeletal symptoms for lab personnel is repetitive work. There is nothing wrong with individual tasks when they are done only a few times or for short periods of time, but as the name suggests, it refers to tasks and actual motion, which is repeated over and over again, easily hundreds of times per day. One of the most obvious repetitive tasks in lab is, of course, pipetting, but also opening and closing of tubes and vessel caps falls into the same category, and you can probably, depending on the lab you are working in, think of other examples as well. Repetitive work is typically something that cannot be avoided, but what you can do is to understand your own limits and be aware of the risk of repetitive strain injury. Here, the symptoms, which typically include various levels of pain in the wrist, forearm, and hand, develop gradually over time, becoming more severe. And this is why it is important to be mindful of the work you do and take precautions to prevent any injuries in the first place. It has been observed that even quite short pipetting times are associated with hand pain, which is of course in itself very tiring to a person. The graph here shows percentage of lab workers and complaints about their hands categorized by the minutes they pipette. You can see that hand complaints increase over time, and already after 30 minutes of daily pipetting, quite many experience some strain in their hands. And we all know that longer pipetting sessions are extremely typical, and after only 30 minutes, you might not even be half done. So what then is a safe level of pipetting? This is of course very individual, and there are no generally accepted recommendations or guidelines for this. But of course, if the feeling of pain or strain doesn't go away during rest, the duration and continuous pipetting should be decreased, and definitely better ergonomics should be looked into. But luckily, there are many things that you can improve in everyday work in the lab. Some of these are smaller things, but together all these individual elements make the overall ergonomics much better. Workplace design is one of the obvious ones. You should always make your own workplace as optimal for you as possible. Have the equipment and consumables that you are using as close by as possible. And for example, those reagents that you need only occasionally a bit further away, but still well within the reach. Always consider how you work and what the flow of a given application is, and plan and design the layout of the workbench accordingly. Personal awareness is critical, as ergonomics is brought into action by ourselves. Be mindful of your posture, like I am now, and for example, try to avoid reaching out towards your samples with your neck like this, which is very typical for anybody in the lab to do. Training, that's a very important part. You can't improve something that you are not aware of. So you have to kind of think, what is the most ergonomic way of working, and see how you could improve that part in your daily life. And then, of course, there is the tool selection. With proper tools, you will not only have better results and better ergonomics in general, but you are able to avoid unnecessary repetition, which will in turn reduce strain. Here we will focus on pipettes, but this same sentence is true for any lab equipment that you might be using. So, how to select an ergonomic hand tool? Here, a pipette. The size and weight of the pipette is of course one of the main characteristics that contribute to good ergonomics. A more lightweight pipette is easier to use and for extended period of time, and a short length pipette gives you an optimal shoulder and arm posture and a good reach to different tubes and vessels. It is actually equally important that the pipette sits comfortably in your hand, and a correct pipetting position in hand is found intuitively. Having an ergonomic finger hook in the pipette is extremely beneficial, as it helps to optimize multiple holding positions, and the wide surface area distributes pressure to a larger area in your hand, making the use of the entire pipette much easier and lighter. In addition, you shouldn't have to look for behind the pipette for the volume settings if you are, for example, left-handed. Overall, the numbers in the pipette should be big enough so that you don't need to change your posture and bend closer to have a look at the volume settings. The volume information should be available to the user so that the normal neutral posture is supported, and you can simply check by a quick glance in the direction. Also, the volume adjustment itself and the pipetting and tip ejection forces should be doable with a minimal force. One thing quite a lot of people miss when they choose their laboratory tools is how easy it is to maintain them. Lab equipment, including pipettes, are highly sophisticated and they do require maintenance. You need to clean them on a daily basis on the outside, but also regularly on the inside. But if your chosen tools are difficult to disassemble and maintain, it is less likely to be done. You are more likely to end up sending your pipettes for maintenance every year without cleaning them on the inside yourself, because the whole operation is difficult and might even require some special tools. This is an issue because poorly maintained instruments will make their use less reliable and also heavier. For example, when any kind of dirt accumulates in between the buttons, it will make the use of those buttons much heavier. So, when designing Sartorius pipettes, these ergonomic factors that I just described are always kept in mind with the design and functionality for the pipettes. The pipettes are very lightweight, and always the shape and function of the human hand is considered with the design. The pipettes are designed for both right and left-handed users, which is something that I personally have really come to appreciate as a left-handed person after spending years of checking from behind of the pipette what again was the volume that I just adjusted. These pipettes have the ergonomic finger hook that I just mentioned, and they rest very easily and comfortably on your hand. And then any kind of muscle power that you need for adjustments or pressing the buttons or loading or ejecting the tips is it can be accomplished with very minimal muscle power, which is of course very nice. As an example that I will also discuss in the following slides, I'd like to introduce a few of the pipettes. Here is the electronic one, Picus, on top of the slide, and here is its mechanical counterpart, the Tacta. Then one thing to also bear in mind is that how you actually choose your pipette tips is not a trivial thing at all. The pipette and the pipette tip form together a system where the design and function of both parts is equally important. The standard OptiFit and filtered safety space pipette tips are ideal for use with Sartorius pipettes like the Picus and Tacta. They enable maximal accuracy and precision and a good seal between the tip and the pipette. But in addition, they also ensure comfortable ergonomics because loading and ejecting the tips is as easy as possible. And you do not need to use extensive force to attach the tip or to eject it. And of course, with the more reliable and accurate results, you end up doing less experiments and less repetitions in the first place. So, let's take a few moments to compare pipetting forces required by different pipettes. There are significant differences between pipettes when it comes to operating forces. A single pipetting cycle, of course, includes the aspiration and dispensing of the liquid, and then finally the blowout and tip ejection. The overall force required for these individual steps is quite modest, but these are again something that accumulate over many repetitions. If you look at the comparison here between different pipettes, you can see that there is a difference which is quite big. If we look at Tacta, the mechanical pipette, you can see that it requires almost 40% of less force to use compared to some of the less ergonomic alternatives, which are here marked in gray. So think about how much of a difference it makes when you multiply this by hundreds of repetitions you actually do with the pipette on a daily basis. When we then compare to Picus, which is on the far right in the picture or in the graph, the difference of force required are even more obvious, with up to 85% of less force required with one pipetting cycle. Tip ejection is the single most force-demanding individual step during pipetting. So electronically controlled ejection or then a very soft tip ejection will save you from a lot of strain. And as we have discussed, when pipetting tasks are repetitive, your exposure to awkward postures and applied forces can increase your risk of injury over time. To mitigate this risk, it is good to consider using an electronic pipette where possible, because this requires similar amount of pressure as, for example, your laptop keys, or then as ergonomic manual pipette as possible. Then, of course, use of multi-channel models reduce repetition, as you can simply go with 8 or 12 channels at the same time. We have talked about the ergonomics and forces required by pipetting, but let's also take a few moments to consider the end result, the quality of pipetting and the pipetting results. Here we have tested a couple of pipettes on the market, and in the test setup we have had participants pipette for 3 hours straight. And we measured here the pipetting performance and at different time points. And here, as you can see, the Tacta, the mechanical pipette, in yellow, and the Picus, the electronic one, in blue, here in the picture. With ergonomic pipettes, such as Tacta and Picus, the standard deviation of the pipetting results was quite stable, as you can see, and with even a decreasing trend over time. With the electronic pipette, the Picus, the results are even better as the user doesn't tire their hands, plus the electronic control always improves pipetting results even for more skilled users. With the less ergonomic pipettes, you can see that the user gets tired over time, and this affects eventually the quality of results. If the pipette is heavy to use, the quality of the results suffers. This can then in turn lead to even more repetitions, because you might have too much unexplained variation in your results, and you might end up doing even more repetitions because of this. The pipette in the lab can be thought of, I think, as an extension of your hand, and it should be definitely also be feeling like such, so that you can have a good and reliable flow of work, especially with the longer pipetting sessions. So, if you want to learn more about pipetting and related ergonomics, I can recommend Sartorius Pipetting Academy for you. It has a variety of certified trainings on all pipetting related topics. The content is a wonderful source of knowledge to anyone in a lab, and a definite must for the newcomers, the students, but also very, very good for more experienced lab professionals. So I challenge you to go and have a look. It is a great way to improve your skill sets even further, and perhaps to discover new golden nuggets of knowledge. The modules here, as you can see, are a good balance of theory, and also there are a lot of hands-on sessions that you can try yourself. If we think about sample preparation for any experiment, it often is one of the greatest sources of variation. So taking care of and maintaining good pipetting practices and mastering different techniques really makes a big difference in the end. And here in Pipetting Academy, we also have one dedicated session module on all things ergonomics. So this will give you a lot of more content and everyday advice for making pipetting and laboratory work more ergonomic. So go and have a look. So then, to summarize the main points that I have talked about, you should be aware of the importance of managing ergonomics because it benefits everyone, both the workplace and you as an individual. For your lab, good ergonomics support productivity and quality of work. As we have discussed, the quality of results is related to ergonomic well-being of individuals, and it can really be improved with proper equipment and by taking care of the well-being of the people who actually work in the lab. You can improve productivity through reducing the amount of sick leaves the staff members might need due to strain coming from different lab-related work tasks. For the individual, the benefits of course include staying in good health, which will then support long-term working ability, not to mention the motivation and commitment to your work. And obviously, this will make your work more enjoyable in everyday life, because it is, let's face it is much nicer to start your day knowing that it will not end up in a headache at the end of the day. So, to conclude, remember, be aware and recognize your personal risks and which tasks in a laboratory are straining for you. When pipetting or doing other lab work, remember always your posture. Again, take breaks. Those are excellent things. So go take a coffee break, go talk with a coworker, and take shorter breaks just to stretch and give your hands a few moments to recover. Proper tools are extremely important, and this should not be overlooked. Pipette, for example, is an extension of your hand in the lab, so it should be something which is lightweight and easy to use. And always bear in mind that at the end of the day, you have to take care of yourself, because that is where it all begins. And with that, I say thank you.
Hi everyone, I'm Nadim Mira, Product Marketing Analyst at Waters Corporation. First of all, I would like to thank Maria for her nice talk and for sharing all this valuable information about pipetting ergonomics and Sartorius solutions. In the second part of the webinar, entitled Automated the Tedious, Focused on Safety, I'll be talking about ergonomics and how they apply to automated pipetting, the choice of the ergonomic pipetting tools, and some ergonomic considerations when using Pipette Plus guided system and the Endo Plus pipetting robot. Automated pipetting is fundamentally a more ergonomic approach than manual pipetting. By integrating liquid handling automation into routine pipetting procedures, this transfers repetitive, tedious, and time-consuming pipetting steps to the liquid handling robot. This reduces the risks of repetitive strain injuries, especially when pipetting a large number of samples manually over an extended period of time. It frees up time spent on manual labor for high-value activities, and finally allows operator to vary motions by adopting a more dynamic posture, changing tasks, and even taking breaks. However, there are still some ergonomic issues that need to be addressed. First, to select the most appropriate ergonomic pipetting tools. Secondly, to choose the right level of automation to fit your application. And third, to consider ergonomics in guided, semi-automated pipetting and full liquid handling automation. Let's start with a selection of the appropriate ergonomic pipetting tools. For reliable automation requires reliable tools, and for this we have chosen the market-leading Picus pipette from Sartorius to use with our liquid handling solutions. The hardware of the Waters connected electronic pipettes, it's not so different than that of the original Picus pipette, using the same user interface and sharing similar properties, ensuring first superior ergonomics, guaranteeing accurate and precise pipetting results, and finally offering high flexibility, covering a wide volume range from 0.2 microliters up to 10 milliliters. However, in contrast with the original Picus pipette, the Waters pipettes have an embedded Bluetooth capability that allows for wireless two-way communication with the OneLab software. This capability enables a remote pipette programming through OneLab. In other words, pipetting parameters are defined upfront in the OneLab protocol. Then, commands are sent via Bluetooth to the pipette to automatically set defined parameters while the operator or the robot is conducting the experiment. Secondly, a step-by-step recording that ensures full traceability of pipetting actions throughout the experiment. Within the connected environment of the OneLab software, the Waters electronic pipettes can be either used manually without automatic guidance and control from OneLab, or with the Pipette Plus Smart Stand for real-time guided pipetting and traceability through OneLab, or with the Endo Plus pipetting robot for fully automated pipetting and traceability through OneLab. It's important to note that ergonomic risks are significantly reduced when using the electronic connected pipettes manually or with Pipette Plus, since no significant effort is required to changing pipetting parameters from one step to another, but are completely eliminated with the use of the Endo Plus robot. Then comes the questions how to select the right level of automation. In the lab, manual pipetting, guided semi-automated pipetting with Pipette Plus, and fully automated pipetting with Endo Plus can be used effectively to achieve the goals involving liquid handling. And these practices, working together harmoniously, can benefit today from the connected lab offered by OneLab to ensure full traceability and reproducibility. However, the pipetting setup to choose will depend on the application and level of performance to achieve. In the following comparative table, it's listed several key factors to help an operator decide which method is best suited for the application. And among these factors, you can see that we have the risks of repetitive stress injuries that need to be evaluated. Manual pipetting is fast to apply and only requires the skill and agility of the operator's hand to manipulate liquids, respecting lab work ergonomics and safety instead of extra hardware to deploy. It's suitable to process a few samples in a low throughput context. Using Waters pipettes, just like the original Picus pipette, setting up pipetting volumes is digital but done manually. High reproducibility is not fully ensured and can vary between operators and from lab to another. Here, labor cost is not a concern. The operator is subject, however, to ergonomic injuries and also biohazard risks. Applications are limited to low-level preparation, such as coating and dried reagent reconstitution. While manual pipetting is straightforward, it's the main source of variability and still introduce ergonomic stress. Pipette Plus offers a way for a lab to scale up productivity and increase reproducibility by guiding the operator throughout the experiment and automating some aspects of liquid handling. It's very helpful to process several dozens of samples in low to mid throughput level. Reproducibility is ensured as well as traceability. Labor cost is of a moderate importance. The operator still handles the pipettes manually, but ergonomic risks are greatly reduced. So instead of the lab technician manually setting volumes on the pipette while at the same time keeping track of steps in the protocol, Pipette Plus automates these aspects of the work, switching settings instantly from one step to another, only requiring the technician to move the pipette from one labware to another following the instructions on the monitor. And then it's only a matter of a single click to execute the pipetting action. Applications include mid-level sample preparation such as serial dilution and concentration normalization. EndoPlus is most valuable when a more advanced level of automation is required, with the possibility to completely remove user interactions with the system by implementing connected devices and tools such as plate gripper to fully automate the workflow. It becomes valuable when the number of samples increases to a couple of hundreds, targeting a mid to high throughput level. Automated pipetting further improves reproducibility and lab productivity. Here, pipette volume settings, just like with the Pipette Plus, is digital and automated. By removing user interaction during pipetting, the operator is no more subject to ergonomic and biohazard risks. Applications include more complex workflows, such as glycowork and glycan sample preparation, DNA media prep, antibody bead purification, and solid phase extraction. When it comes to using Pipette Plus, just like manual pipetting, you should be aware of ergonomics to consider. You must adopt a balanced body position while sitting on the lab bench to reduce strains on the muscles, tendons, and ligaments, and to maintain the vertical alignment of the head and the spine, as shown in the reference figure on the right side of the slide. You must place items on the working bench according to the importance and frequency of use to avoid excessive range of movements and tasks that require excessive reach. By organizing and optimizing the workspace, you promote ergonomics and safety during pipetting. As a reference, you can see on the visual how items are arranged on the bench to ensure easy reach while maintaining good posture. Labware, tip box, and Pipette Plus stand are inside and at the upper limit of the primary zone for easy, frequent reach. Benchtop, waist, is in the secondary zone for occasional reach. This also keeps it away from the primary zone to avoid contamination while discarding tips. Tablet showing one lab for guidance is within the tertiary zone for rarely needed reach. And finally, as an example, the microplate Peltier Plus is at the upper limit of the secondary zone for occasional to rare reach during the experiment. Using Pipette Plus, the operator will benefit from real-time, step-by-step guidance and automatic control of pipetting parameter, instantly changing volume and action from one step to another without exerting significant force. Moreover, the use of the multi-channel pipettes helps reduce the repetition. Now, EndoPlus is an open liquid handling system, so no opening and closing of a front door is necessary to access the working deck. It has a small footprint and a compact benchtop design, which makes it easy to set up and manipulate. Introducing EndoPlus into your routine lab work will help eliminate repetitive, tedious pipetting, thereby preventing ergonomics injuries. Frees up time for the scientists to focus on other creative, development-oriented tasks, and improves lab productivity with collaborators not losing much time away from work due to repetitive stress injuries. So with EndoPlus robot, there are some ergonomics that were taken into account to ensure an optimal operation and use of the system. EndoPlus provides a large flexible working deck for optimal viewing and easy installation of magnetically attached labware holders known as dominoes and as well as connected devices. It uses visual deck layout and numerical labeling to help install dominoes and devices in the correct positions in a smooth and effortless way, and to rapidly switch between configurations between runs. Finally, EndoPlus offers easy access to the working bench with no need to bend forward to reach items when it is placed on a steady table at the optimal working height, just like you can see in the photo on the right of the slide. where Endoplus is placed in the hood, with the lab technician installing the consumables in the dominoes. So the take-home messages are: ergonomic injuries are not only a risk for operators that perform repetitive manual pipetting, but can also affect operators using semi-automated and fully automated systems. To reduce exposure to repetitive stress injuries, it's important to evaluate the task to ensure using the appropriate pipetting using the appropriate ergonomic pipetting tool. For example, electronic single channel versus electronic multi-channel pipette. And secondly, selecting the right level of automation. Here, Pipette Plus versus Endro Plus for full automation. Endro Plus can not only improve reproducibility and increase lab efficiency, but also greatly reduces the ergonomic risks in the lab, as well as biohazard risks when placed inside a fume hood. In the third part of the webinar, we are glad to present the testimonial of Vannes van Holm, a PhD researcher and user of the Pipette Plus and the Waters connected electronic pipettes at the KU Leuven that was interviewed by my colleague Jasper Eaton, and explaining the reasons that motivated his lab to look for automated and similar solutions to reduce the impact of repetitive stress injuries on staff health and lab productivity.
All right, so hello, Wannes van Holm. Thank you for joining us for this interview session. I'm really glad that you could be here with us, and I'm sure our listeners will really enjoy the feedback that you have to give on this topic about ergonomics. So, first of all, who am I talking with today? He's a soon-to-finish PhD student specialized in periodontology and oral microbiology. So, from the Department of Oral Health Sciences at KU Leuven in Belgium. We do have a couple of questions for you. So again, thanks for your time, and we can get started. So maybe first question with a quick presentation. You probably do this all the time, but a quick presentation about your PhD topic, what you do specifically.
Yeah, so oral microbiology, we're very interested in what's happening in the mouth, and more specifically, we wanted to see, okay, healthy people, unhealthy people, what kind of microbes do we have in the mouth, understand which are associated with which kind of disease, and then see, can we specifically target the ones that are associated with diseased people, and then also benefit the good ones so that we can restore a balance between the oral microbiome and then have hopefully healthy and happy patients. That's the core of it.
Very broad topic that seems to apply to everyone, right?
Yeah, I like to generalize.
Yeah, of course. No, that's the right way to put it. So just follow-up question to that naturally is, of course, you're sitting in your lab there at the university, and maybe we can get right down into the details about what applications and specific workflows that are routine in your lab, or that you do often, not necessarily routine, but that you regularly use pipetting for.
Yeah. So I pipette daily. I have to make enough notes, so I write a lot. But I think even more than writing with pen and paper, I pipette even longer each day. So very routine. And I think many people will recognize if you do it that much, you start feeling it. And some evenings, it's harder to do something else. And so what are the things we most of the time do? Liquid handling with bacteria in it, and then because we want to know what those bacteria are doing, we then further process them through flow cytometry or extract their DNA to look at it through qPCR or sequencing, and especially those last two can be very intensive with repetitive steps.
A lot of pipetting, so mostly dilutions, transfers, and a lot of that.
Yeah.
So a lot of plate work, which multiplies out the pipetting steps necessary for the most part.
Every sample exponentially increases the workload and how tiresome it is indeed.
Right. Okay. Sounds like a lot. So I remember initially we were talking about automating those types of steps.
Yeah.
What motivated you specifically? So just what you said here, but maybe there are other factors, maybe more specific, that motivated you to look for automation solutions or similar solutions to optimize those types of workflows in your lab.
Yeah. So it's very hot topic to be like very high throughput.
Right.
But for us, yeah, okay, we can do it quickly, we can work harder, but the high throughput wasn't the main focus. It's always nice to use a buzzword as high throughput, but something that I feel is overlooked and sometimes underappreciated is what it does to people. So we have a very essential part of our lab, a lab technician who has pipetted for 25 years, and she almost had to stop with lab work, even though she's not even close to her pension, or getting there, but still very valuable.
Still not close.
Purely because of repetitive strain injuries, and yeah, she couldn't eject tips anymore, so she had to actually stop from the doctor. She wasn't allowed to enter the lab anymore for the risk of making it even worse. So we were looking for solutions. Okay, can we reduce this stress? How can we make a functional or like an essential part of our lab functional again? So that's why we looked at a bunch of different options, like automation and also ease of use through pipettes.
Ease of use through just standard pipettes in the end, because you didn't end up opting for the kinds of automation that are out there on the market, whether it's low throughput, benchtop types of systems, or even the higher throughput, as you were using that key word. Even those higher throughput systems weren't adapted here specifically. And actually, in the end, you decided to opt for the manual electronic pipettes from Waters to perform those pipetting tasks instead. So that it's quite interesting to find that people that are going towards automation at the end of the day don't necessarily always need that. Would you be able to describe how specifically those pipettes have impacted your work and the main advantages that you've seen when actually using them? So whether it's you or your colleague that experienced pretty intense repetitive strain injury in this case.
Yeah, so indeed, we were initially also looking at using a robot, but due to the moving around, because we were looking for options throughout the whole lab, just not in one area, and so moving a whole setup would be difficult. And especially, we are quite a big university, but space is always limited, and the footprint of I think the Andrew was quite low, but still
there, still present. Yeah.
Just running around with the pipette is much easier. So that was our main idea, to just see can we reduce it. And so we looked at the pipettes, and when we got them, the first thing we noticed immediately was just the pipetting, so much more comfortable, ejecting tips so easy. It's hard to put in words how much it changed. An anecdote I can give you is that maybe she will pass through in a bit, because the qPCR is running over there.
The machine is right there.
Yeah, she put, there are two actually. She put them both in, from going no lab work to the same throughput as years ago. It's like almost as if, of course she still has some issues, but it's right. The time, the clock has turned back for her wrists. So that was like the craziest thing. But secondarily, when she's not using it, I tend to steal them for myself. And they're like functions like the multi-dispense, but even just the comfort of using them saves so much strain. And if you're doing this for a few hours, the strain makes you work slower, and you make more mistakes. But if you don't feel the strain, you can work harder and quicker. And like many of the things that took way longer before feel so much comfortable, and it's
more streamlined. So even just basic things like automatic tip ejections and just the lightweight ergonomics of an electronic pipette, even if being done manual.
I was worried that they would be slightly heavier, but absolutely not. They are comfortable and pleasurable, fun to work with, and even have fun feedback noises, which make it
right there. Ding.
Yeah.
Yep, absolutely. Well, I mean, thank you for that account. I guess there's always a flip side to a coin, or something that people should be aware of when looking to try to simulate or do the same types of work as you and purchasing electronic manual pipettes? What should future customers look out for? What do you think, in your opinion, future customers should look out for when looking to involve these in their lab?
I wish I could be more negative to give a more realistic vision of it, but we have no regrets on buying these. In the very beginning, there are a bunch of options that have some learning curve. I use more of them than our older colleagues, but just the normal pipetting tasks are so much easier, so much more comfortable. And so, okay, the main issue that we had faced first is the purchasing cost, because it's, of course, a little bit more expensive.
Right.
But we do not regret our purchase, because, yeah, we did it to save our colleague, to save myself, and a lot of strain and injury. So maybe...
And it ended up paying off.
Yeah, it's paying off. The only caveat I have is that maybe don't give it to students, because clumsy students are the bane of a lab. But yeah, we are very happy with our purchase.
That's good to know. So people should, as you mentioned, just look out for the initial price point and see if you really get a return on investment over your time. In your case, visibly, it was made, that return on investment.
If you use it as much as we do, and you go home feeling the strain in your wrists, it's such a saver.
Right. And the other thing just to look out for, as you mentioned correctly, is just the slight learning curve that's associated with just getting comfortable using these.
If you want to get everything out of it, yes, there's a learning curve. If you just want to do the basic tasks, already that alone is
already a win.
Yeah, exactly.
Well, thank you very much, Wannes, for your time and the answers, well described answers to our questions. Really appreciate you spending some time with us. I hope our listeners managed to get some interesting feedback here, and we'll leave you back to writing your PhD. That's almost done. Best of luck with that, and thank you again.
Super.
All right. Well, thank you very much to all of our speakers here. Great. We've got everybody back on camera. Awesome. All right. Well, I want to thank you again, Maria, Nadim, Jasper, and Wannes, for your time in your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. You've got lots of great questions coming in from our audience here. And this first one, I'll pass over to Maria to kick us off. So Maria, this first question asks, what kind of certificates and purity level you have available for the pipette tips?
Okay, a very important question for anyone working in the lab. Well, this depends a little bit on the product, on the tip. So we have either pre or non-sterilized tips, for example, and then majority of our tips at Sartorius are also certified to be free of DNAs, RNAs, human DNA, and endotoxins or pyrogens.
Great, thank you. Now I have a question for you, Nadim. This first question asks, can Andrew Plus fit into a fume hood? And are there any special requirements for operations in fume hoods?
Yes, it's a very good question that we keep having from our users. So the workbench. Let's start with the system itself. So the workbench of Andrew Plus is modular, as I said in the presentation, based on the experiment requirements. In other words, Andrew Plus can use 1, 2, or up to 3 rows of dominoes and connected devices. The third row configuration counts for a total of 11 dominoes. So one should make sure that when they would like to set the system in the hood, that the inner surface, they should make sure that the inner surface of the hood is large enough to host the system with the required number of dominoes devices used by the Andrew Plus. So this depends on the workflow. So this is the first requirement. Something important to consider also is that the fume hood should be able to support the weight of the unit, which is around 16 kilograms, without being bent. So the surface of the hood must be really very stable and flat, steady and flat, and ideally in one piece. Because if any distortion happens to the surface, this can impact the XYZ calibration of the robot and therefore impede the handling precision. So this is a challenge that we already saw in some type of hoods, and we try really to give their good recommendations and guidelines to our users when setting up the system in the hood. Just for information that we would like to, if not, we are already working towards proposing for our customers and users a custom-made cabinet, specially designed for the Androplus and connected to OneLab to further monitor and for further monitoring and traceability. For instance, collect information about the temperature, the humidity, and many other parameters that can be involved. Regarding the requirement of installing Androplus in the hood, and are present in the pre-installation checklist that you can find in our help center. So Kylie, I don't know if you can, we are able to share the link with the audience. So this is an article from our help center that you can go and check. So there's several documentation there about the Androplus. Thank you,
Great,
Kylie.
thank you.
Thank you, everyone.
Thank you so much. And that link has been sent, so you should be able to see that, our audience there on your screen in that Q&A window. All right, we've got some more questions in here and not that much time, so we want to try to get through a few more. This next one, Maria, I'm going to pass over to you. This question asks, I am looking to reduce the amount of plastics in the lab. How can I do this when using pipette tips?
Okay, well, that's a very, very current and important question, actually. Well, if we consider single-use plastics, such as the pipette tips, this can seem a bit challenging, of course, but there are definitely things that you can do. You could, for example, choose different packaging options for the tips, those ones which have less plastic, such as the refill or bulk products. So these are the products that actually come without the tip box. So this using these products allows you to reuse some of the empty tip boxes that you might already have in your laboratory. And if we are then, for example, thinking about the number of repetitions that you need to do for each experiment, of course, so time-wise and tip-wise, it would be nice to avoid any unnecessary repetition. So choosing high-quality tips for your pipette means that you will have more accurate and repeatable results, which in turn means that you are doing less repetitions and using less tips. And then, of course, like overall planning your experiments well, so that you are using as little tips as possible will help.
Great, thank you. Now I have another question for Nadim. This question asks, is OneLab compatible with LIMS or ELN?
Very good question. This is related really to the connectivity that we have around OneLab. So we currently do not have one seamless solution to connect OneLab with LIMS and ELN. Presently, the import and export of information functions through a simple data file, like an Excel sheet. This can serve as a link with the actual LIMS and OneLab. Any request of integration with specific LIMS will require a custom development on our side, and this will be processed case by case. With that being said, we have already developed and implemented a solution to connect OneLab with some aspects of LIMS, which is a new tool only available on the enterprise private deployment of OneLab. And this tool is named File-Based Association, that allows user to automatically generate one or multiple pending experiments by importing one or multiple work lists into the dedicated folder within the private OneLab server in-house, if it's deployed in-house. So I think this is the solution that we have so far, and we can also share here a valuable link which can be helpful from our help center about how to configure the file-based association system with OneLab.
Perfect. Thank you, Nadim. We sent that second link out as well.
Thanks.
And for the sake of time, we do have so many more questions that need to be answered, but we do want to make sure that we can provide our final comments here and wrap up. So for the sake of time, we will be closing the Q&A session at this time. So I want to thank you again, Nadim and Maria, for your time today. But do you have any final comments for our audience here before we go?
Yeah, sure. Shall I, Nadim, go first? Well, first of all,
go ahead, Maria.
Yeah, thank you. First of all, thank you for the audience. It was great to have this opportunity to discuss about ergonomics. I really hope you enjoyed the webinar. Thank you, Nadim and Jasper. A pleasure as always. And thank you, Kayla, for hosting us. And as I already mentioned during the presentation itself, please go ahead and have a look at what we have to offer at the Sartorius Pipetting Academy. It's definitely a treat for anybody working in a lab. Thanks.
Thank you, Maria.
Same on my side, maybe. Kylie, just a couple of words. First, I would like also on my side to thanks everyone here, all the audience that stayed with us until the end of the webinar for the questions, and for the also organization, Kylie. So just in 2 words, I really would like to invite you to participate to our live webinar series that are presented by Jasper Eaton. So it's really, if you're interested in knowing more about our solutions, this is a really good series talking about the connected lab and how to overcome and mitigate challenges and risks. So far, there's in total, there's 4 episodes besides the introduction. 2 episodes were already broadcasted that you can watch again, and the two others that will be coming in the next month and in November. So you have the link at the bottom of the page, and you have the QR code that you can scan. It directs you to the page on the IndoAllianceWaters.com website. So that's all my side. And thank you again for everything.
Wonderful. All right. Well, thank you so much, Nadim and Maria, and the rest of our speakers for your time today and your important research. We would also like to thank Labroots and our sponsor, Sartorius, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. Now, this webcast can be viewed on demand, so Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Thank you, everyone. Bye-bye.
Bye.